So I'm just going to record this. So the reason I'm going to go through some of these today is fact that let me just sort out my screen. Basically, I'm going to go through these because some of these can be quite hard to answer in the exam questions. So what I'm going to do is I'm going to go through these ones now so that yeah, we've got a bit of a better idea of what we need to do when we answer these questions for protein synthesis, because some of them can be a bit tricky. So we get a question like this. So they seem quite simple. They're 3-mark questions, so in total it's 6 marks. And you're asked to describe the role of messenger RNA, so that mRNA that we've done lots about the past few days, in the following processes. So first of all, you're asked to describe it in transcription. So we sort of basically want to say, well, what does messenger RNA do? What is its role in transcription? Well, the first thing that it does, that it is really, and this is probably the most important thing about messenger RNA, is the fact that it is a copy of the DNA. It makes a copy of the DNA. So that's that first thing that we're going to want to say, is we're going to say it is a, oh, my writing has gone very big. Let's put there. It is a copy of the DNA. So it's a copy of the DNA. That's the first thing we want to say. It's a copy of the DNA. That would get you 1 mark. So if that's all you can remember, oh, my pens are going to be huge, aren't they? I don't know what's happened to them today. If that's all you can remember, you would get 1 mark for saying messenger RNA is a copy of the DNA. You get another mark if you actually said it was a copy of part of the DNA. So if we expanded this answer actually, and we said this, mRNA is a copy of part of the DNA, you would actually get 2 marks for saying that, because you've actually expanded on that answer there. So saying it's a copy gets you 1 mark, but saying it's a copy of part of the DNA gets you a 2nd mark. Other things you can say is you can say it's a copy of one of the strands of DNA. So that would get you a mark. So it's a copy of one of the strands of the DNA. So a full answer that you would expect to see somebody write, I'm going to write one up now. If I can get rid of this. Oh, here we go. I have got an eraser. There we go. Right. So a really perfect answer that would be really nice to see on a question like this would look something like this. So we would say, so mRNA is a copy of the sense strand. Now remember, the sense strand is the coding strand, is a copy of the sense strand. Sense, and here I'm just going to put coding. mRNA is a copy of the sense strand, the coding strand of DNA. Now we need to, oops, what have I done? Of DNA. We need to include the fact that it's of DNA there. That's important. But that isn't going to give us a whole 3 marks. So that would give us the first part. So mRNA is a copy of the coding sense strand of DNA. We then could add something along the lines of, I don't know what is happening to my screen. We could say something along the lines of mRNA is complementary, so complementary base pairing, to DNA. or specifically this would be nicer, is mRNA is complementary, base paired to the template strand. A nicer answer. mRNA is a copy of the sense strand of DNA. mRNA is complementary base paired to a template strand. Another thing I would always want to sort of include, I'm going to pop it here, would be that mRNA carries the genetic code. mRNA carries the genetic code to the cytoplasm. So obviously the DNA can't do that. Okay, so it's a copy of it, complementary base paired to template, and it carries it to the cytoplasm. So then we get the second question. Okay, so what is the role of mRNA in translation? So let's do this one in a different color just so we can see it a bit differently. So, well, what is its role? Well, this time it does something very different. The genetic information, the base sequences, the codes in the mRNA determines amino acid sequences. So what have I done? So let's start. I'm pressing on something on this screen, that is for sure. I'm just going to move this off a minute. Screen a little bit crazy. Okay, so we could say something along the lines of the codons on the mRNA codons bound on mRNA determine the amino acid sequence. It complementary base pairs it being the codons, so it will be the codons complementary base pair with anticodons on tRNA which carry amino acids. In this situation, that would give you 3 marks. I'm going to highlight where you get your 3 marks. So by saying we've got codons, that gets you a mark straight away. So the codons are found on the mRNA. They determine the sequence of amino acids. That gets you a mark. And by saying that it complementary base pairs with the anticodons gets you another mark. Other things you could say in this situation here, anything that talks about stop or start codons, so stop or start codons. So you could say mRNA contains stop and start codons to initiate and terminate translation, or anything along the lines of mRNA binds, mRNA goes to a ribosome for translation to take place. And again, the marks that you get here is just my mention start and stop, and the word ribosomes in the right context would get you the mark there. So that's that first part. Let me get you the next question up. And this is where they can get a little bit tricky. So here we go. Here's the next question. So the diagram below represents a structure of transfer RNA. Transfer RNA has a similar structure to messenger RNA, mRNA, but the single strand of nucleotides is folded back in itself and folds are in place by base pairing. So your questions are: list the 4 bases in the region labeled A, which pair with those already shown. So you're basically just asked to do basic complementary base pairing. So you've got GGAA there. So what you'd expected to do is put C. Now remembering this is tRNA, not DNA. So then you would want to put U for uracil, and that would get you the 1 mark there for that. Then you're given this B, the 3 shaded regions in region B. So shaded bases in region B, that's what we want to pay attention to, are responsible for binding to mRNA. So they bind to an mRNA molecule during translation. Give the name used to describe this group of the 3 bases on the tRNA, and we know that is an anticodon. Now, each amino acid is coded for by 3 bases on the mRNA molecule. Explain why a group of 3 bases is the smallest number that can be used to code for all amino acids found in proteins. Here we go. This is a really good question. I've lost how many marks that is, so let's have a quick double check. That is a 3-mark question. So let's go back. What are we going to want to say for this? Well, the first thing that we're going to want to mention somewhere is we're going to want to use that keyword degenerate. We're going to want to include that somewhere. So basically, we want to remember that when we use, so we've got 4 bases. So I'm going to give you a really good example answer. So basically what happens here is, so explain why a group of 3 bases is the smallest number that can be used to code for all amino acids found. Okay, so it's the lowest number when 4 bases are used that give enough permutations Permutations is just basically the amount of combinations we can get. Enough permutations to code for the amino acids, to code for the 20 amino acids. So, lowest number. So we want to put a little bit of maths in here. So we would go, okay, well, we've got 20 amino acids. We've said that we've got 20 amino acids, actually, so that's fine. I don't need to rewrite that bit. So that bit's really important. And we've got the fact that it's the lowest number, which is really important as well. Then we're going to say, okay, well, when we've got 4, lots of bases, and we're going to do them in the triplet code, what we end up, when we do 4 bases in a triplet code, we end up, like we said yesterday, with 64 permutations. So our code, so we end up with more. So we've lowest number when 4 bases are used that give enough permutations to code 20 amino acids. You could actually put in here, so we could say lowest number when 4 bases are used, so the lowest number being the 3 for the triplet. And the 4 bases used that give enough permutations for the 20 amino acids. So 4 lots, so Kent's triplet code gives us 64. We only have 20 amino acids. Therefore, because we have more than that, we can say that the genetic code is degenerate, meaning some codes Some amino acids have more than one code. Okay. Oops, what have I done? And so that is that second set of questions, and that would give you 3 marks. And the important part here is that 64, and then the fact of the use of the word degenerate. And that is, have I missed something? Oh yeah, I've got the word 4 bases there, 4 bases. So there's 5 potential marks there, and it's only a 3-mark question. So let's have another look at a couple more questions. So the particular amino acid bound to a tRNA molecule depends on the 3 bases at position B. So we can see position B there. Why is this important during the translation of mRNA to form a polypeptide? Well, it is important because, let's make this a little bit bigger, it ensures the correct amino acid added to the polypeptide chain. So therefore, we can mention then, all you need to do is mention something along the lines of mention a sequence of amino acids matching the anticodon to the codon. So explain why it's important. So it ensures the correct amino acid is added to the polypeptide chain as the anticodon complementary base pairs. So comp base pairs to the codon, which carries a genetic code. For that protein. Okay, so that's that part of the question. There's one more little bit that I just want to have a quick look at, which is not that. Do I want to look at this? No, I don't want to look at that minute. So we'll leave that for now. What we will do is we're going to look at something not new. We're going to sort of carry on with this topic. But with this topic, we're going to look into mutations a bit more detail. So we started talking about mutations yesterday, but we didn't go into great detail about them. Now we're going to go into a bit more detail about them. So I'm just going to get rid of this. Export that image. Okay, so let's clear this and let's start off with our mutations now. Clear canvas. I'll put my light on because it's gone really dark. So mutations, basically, yeah. If everything went always according to plan, genetic codes would always be perfect. We'd always be making exactly the right protein all the time, there wouldn't be any errors or any issues. However, that doesn't always happen. And mutations can occur. Mutations, we sort of look at them in two ways. We can either go, oh, wow, they're amazing, or oh, wow, they're really bad. And often, they sometimes don't do anything at all. But yeah, in films and stuff, they go, oh yeah, mutations give you superpowers. Often they're not actually really very great. Often it's very rare for mutations to lead to something useful. Obviously they can, because of obviously evolution and survival of the fittest. But in general, when mutations occur, they can be quite harmful. So basically, what is a mutation? A mutation is a change to the base sequence of your DNA. So that is our official, now my pen's gone tiny, I can't keep up with this. So let's rewrite that. So mutations are changes to the base sequence of DNA. That's basically what mutation is. So any changes to that base sequence of DNA from the original, we call a mutation. It can occur in a couple of ways. They can occur due to errors during replication. So they can happen due to errors during replication. And actually the rate of them can change. We can have something called mutagenic agents, and we're going to look at that in a bit. And mutagenic agents can affect the rate. They they can increase the rate of mutations. So the types of mutations that we can get, well, we started this the other day, and let's just have a quick look at the types that we can get. So let's do this here. So our types of mutations. Types of mutations. We've got a list of 6. We did 3 of them the other day, but we've actually got 6 that we need to know. So first one we did the other day, and that is substitution. So substitution is where one or more bases is swapped. And they're swapped for one or the other. So for example, we would have A, T, G, C, C, T, and it becomes A, T, T, C, C, T. So that is an example of a substitution. One or more bases is swapped for another. So G here has been swapped for T there. So that is a substitution. We can have a deletion. This is when one or more, so it doesn't just have to be one or more bases is removed. So then in that instance, we could have A, T, G, C, C, T. And then in that instance, that will become A, T, C, C, T. So we've got that G missing completely. We can have the exact opposite of deletion, and we can have an addition. And you've guessed it, one or more bases are added. And in that situation, we're going to go A, T, G, C, C, T, and it's going to become A, T, G, C, 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 T. So we've added 3 extra C's in there. We can have duplication. Duplication, again, is one or more. One or more bases are repeated. In that instance, we could have something along the lines of A, T, G, C, C, T. And it can become A, T, G, 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 oh, G, C, C, T. So G has been duplicated. Technically, my addition I've done up there could also be a duplication. So I'm going to change this one to make it clearer that it's a addition. So we could have A, T, G, A, C, C, T. So to just define those ones a little bit clearer. Duplication, one or more bases are repeated. An inversion, which we also spoke about the other day. In an inversion, a sequence of bases is reversed. This is a bit different. So a sequence of bases is reversed. This one's probably the most complicated of them, and it's not too bad. So again, we'll have A, T, G, C, C, T on here. And that is going to still have the A there, and then everything else is going to reverse inside. So we get C, C, G, T, and then ends in the normal T. So we've got that section that's been reversing. And our final one is translocation. Translocation is a little bit complicated. Basically, a sequence of bases is moved from one location in the genome to another. This could be within the same chromosome, or it could be to a different chromosome completely. So it could be same or different chromosome. So yeah, they are our types of mutations. So again, we have got, oh, let's bring it smaller. We've got substitutions, one or more bases is swapped. Deletions, one or more bases removed. Addition, one or more bases are added. Duplication, one or more bases are repeated. Inversions, a sequence of bases is reversed. And then translocation, a sequence of bases are moved from one location in the genome to another. That could be to the same chromosome or to a different one. Why is mutations an issue? Why are these things happening an issue? Because you know that the order of those DNA bases in a gene determines the sequences of amino acids. So if the mutation occurs in the gene, the sequence of amino acids in the polypeptide that it codes for could be changed. Now, remember we talked about those ones that do frame shifts the other day. So some of them will always then affect everything that happens later. So we've got frame shift ones. Substitution isn't a frame shift, but ones that are a frame shift are deletion is a frame shift, addition is a frame shift, inversion isn't a frame shift, but duplication is a frame shift. Inversion is a complicated one. It can be a frame shift. It's not always a frame shift. It depends how long the sequence is that you're inverting. But yeah, it's not always a frame shift, but these ones always create a frame shift. And that means all the polypeptides after that, all the bases, all the codons after that are all going to be different to what they were initially. Why is this an issue? Why does the sequence of amino acids change and make a difference? Well, we know that this amino acid chain makes a polypeptide. We know that polypeptides joining together make a protein. A change in our amino acid sequence of our polypeptides might change the final 3D structure of our proteins, which could then mean that our proteins won't work. So, for example, we could have a mutation in a polypeptide that makes an enzyme. The shape of the enzyme then might change. If the enzyme's active site has changed shape, this then might stop the substrate being able to form a substrate enzyme complex. Therefore, the substrate can't bind to the active site. Therefore, the enzyme cannot catalyze the reaction. So then that protein is useless because the mutation has caused it basically to not be able to work properly. So that is the dangers we are dealing with when mutations happen. So that's some issues that we can have with mutations. Some mutations can actually cause genetic disorders, and we'll look in detail at them in a moment. So these can be things such as like inherited disorders that are caused by abnormal genes or chromosomes. So things like cystic fibrosis. And some mutations that we have can increase the likelihood of developing certain cancers. So things like the BRCA1 gene increases the chance of developing breast cancer. This is where it gets a little bit more different each time we sort of deal with it. I'm just moving this around so I can see better. There we go. If a gamete, and remember that a gamete is a sex cell, contains a mutation for a genetic disorder or a type of cancer, is fertilized, that mutation will be present in the new fetus formed. So this is a hereditary mutation, and then that is passed on. But not all mutations affect the order of amino acids. So let's have a look why. So we've highlighted that some are frame shifts, some can affect it, but let's have a look at, let's do a couple of examples to show why not all of them affect it. And it's because of that degenerate nature of our amino acid code. So let's pop this up here. So because the genetic code is degenerate, means some amino acids are coded for by more than one DNA triplet. So I'm going to show you an example. So we have got T, A, T, A, G, T, C, C. No. This is our original gene. Okay. So because our genetic code is degenerate, it means that some of our amino acids, like we know, are coded for by more than one DNA triplet. So for example, tyrosine can be coded for by TAT or TAC in DNA. So not all types of mutations will always result in a change to amino acid sequence. So like we were saying earlier, substitutions actually don't necessarily always cause a change in amino acid sequence. So let's have a look at that specifically now. So let's have a look at one substitution. So this is our original gene. Let's do a mutated gene below it, and we're going to have TAC, AG, TC, TT. I'll do that in different colours. So C here is our substitution. That's our mutation that's occurred. As we've just said, if we look at tyrosine, tyrosine can be coded for by TAT. So this would be our first codon. First codon is TAT, and that makes tyrosine, which is TYR. Our next codon, AGT, is going to code for serine. And then our final one, CTT, is going to code for leucine. If we have a look at this new one, let's look at amino acid chain this time. Right, so we've still got leucine, AGT, still got serine. Okay, TAC, that's the only one that's changed. Well, TAT codes for tyrosine, but also TAC does. So this stays exactly the same. TAC, that codes for tyrosine as well. So we had a mutation, there is a mutation present, but the polypeptide chain hasn't changed, it stayed exactly the same. So the amino acid sequence hasn't changed. So there is still a mutation present, but our amino acid sequence hasn't changed. Let's have a look at another example, this time using an inversion, because like I said down here, not all inversions are going to cause a frame shift. So let's look at one example where an inversion doesn't. So I'm going to draw you the original gene. I'm going to have G. T. I'll explain what, I'm just going to do them in different colours so we can see the inversion easier. CT, G, CTT. Yeah, the only reason I'm doing different colours is when I then go to do the inversion, you can see the inversion easier. It's literally the only reason I've done that. Okay, so that is our original gene. I'm going to find, using the genetic code, what amino acids each one does. So we've got GTT, and GTT codes for valine, VAL. We've got CTG. CTG codes for leucine. And then CTT codes for leucine as well. An inversion occurs. What we get is a whole inverted section. So our inverted section is now going to read like this, and you'll be able to see it because I'll show you it with the colours. It's going to be G, T, C, T, T, G, C, T, T. So as you can see, this region here is inverted. Let's have a look what's happened though. Let's look at amino acids. So we've got GTC, GTC still codes for valine. TTG codes for leucine. And obviously CTT stays the same, and that codes for leucine. So actually our amino acid chain hasn't changed again. So this inversion hasn't caused a change in the amino acid sequence either. So we call these, when the amino acid sequence doesn't change, even though there's a mutation, we call this a silent mutation. A mutation has occurred, but it's not affected that polypeptide chain. So we call these silent mutations. So they're two examples, and yeah, it's substitutions, inversions. And it's only some substitutions and only some inversions. Some inversions will cause a mutation, a polypeptide chain change. Some inversions will, some won't. However, all those ones that I said that have a frame shift, deletion, addition, duplication, they are all going to change that polypeptide chain. So let's have a look at a couple of examples of them. So additions, duplications, deletions, like I've just said, will almost always change your amino acid sequence. And that's because they're actually changing the number of bases that are in the DNA code, which then comes along and actually changes the base sequence altogether. So this causes a shift, frame shift in the base triplets that follow. And we're going to have a look at how a deletion could cause a frame shift. I'm not going to go through all of them because they all work pretty much the same. We did look at these yesterday, but if we have this one, we've got TA. We've got TA, T, T, A, G, T, C, T, T. And I'm going to draw you the mutated gene here. So we've got T, A, T, G, T, C, T, T. So we've got a deletion here, which would be the T and the A that's in the original one. So if I codon these off and get the amino acids they code for, TAT codes, like we said before, for tyrosine. TAG codes for serine. TCT codes for TAT. I've done that wrong. I've given myself one too many of something. That's fine. Let me just delete something. There we go. AGT, sorry, that will code for serine. And then CTT. CTT is going to code for leucine. Now, we've deleted out of here. So what's going to happen is we've now still got TAT, which is still going to be tyrosine. We don't have AGT. This time we've got GTC. And GTC is then going to code for valine. And then we've just got two left on our own. So that's changed that polypeptide chain. So the only other thing with mutations that I mentioned at the beginning that we haven't actually looked at is mutagenic agents. So I said, oh yeah, mutagenic agents, they can increase the rate of mutations. But what on earth is a mutagenic agent? Well, let's have a quick look here. Mutagenic agents. Basically, mutations occur spontaneously. There's nothing directing a mutation, it just happens spontaneously. So that is also really important to remember, is mutations occur spontaneously. Mutations occur spontaneously. So this can happen if DNA is misread during replication. But some things can increase this rate of mutations, and they are mutagenic agents. So mutagenic agents, some examples, we've got things such as UV light, UV light, so UV radiation, UV light, ionizing radiation. These are all examples of mutagenic agents. So again, when people go, oh yeah, the sun can cause skin cancer, it can because there's UV light coming off the sun, and UV radiation is a mutagenic agent, so can increase chances of mutations occurring. How do they do this? So they don't just randomly go, oh yeah, we're going to cause mutations. There's actually reasons why they have this. So mutagenic agents, how do they work? How do they cause mutations? Well, they work in a couple of ways. They can work by, number one, they can act as a base. So what do we mean by that? So chemicals that we call base analogues can substitute for a base during DNA replication, and they change the base sequence in the DNA. So this is what we mean by them acting as a base. So for example, 5-bromouracil. This will take me a second. 5-bromouracil is a base analog. Okay. It's a base analog and it can substitute for thymine. And this would cause a substitution mutation in the new DNA. So it substitutes one for the other. Mutagenic agents can also alter bases. So some chemicals can actually delete or alter bases. So an example of this would be an alkylating agent. And what these do is they add an alkyl group. Oh, no, that's not how I spell alkyl group. That's alkyl. Alkyl group to the DNA, to our guanine in particular. When we do that, that changes the structure. And then that causes guanine to pair with thiamine instead of cytosine. And then finally, 3, it can actually change the structure of DNA. So some types of radiation can actually change the structure of DNA, so that can affect the DNA during replication. So one example of something that can change the structure of DNA is UV radiation. And what that can do, what that does is it can cause adjacent thymine that are next to each other. Thymine that are adjacent to base pair together, and basically that will cause a cleavage. Not cleavage, it will cause a loop where it cleaves in on itself. So what would basically happen is where you would have thymine, and they should go with an A and an A, base pair up, it will actually basically loop around. So you'll have your normal base pairing, and then you'll have thymine here, and then the thymine will loop here and base pair like that, which causes a lot of issues, obviously. So yeah, that is basically the overview of mutations, the issues of mutations. So basically, we've got 6 types of mutations. Some can cause amino acid sequence base changes, and some don't. So these ones, no polypeptide change. Whereas that one up there caused a frame shift. Mutations occur spontaneously, but things such as mutagenic agents can increase the rate of mutation. So things such as UV light, UV radiation, ionizing radiation, and they do this as they can act as a base, and they can basically substitute. So 5-bromouracil is a base analog, and that can substitute thymine. Mutagenic agents can also alter bases, and they can add an alkali. They can be an alkylating agent, which is where they add an alkyl group to guanine, and that causes the structure of guanine to change, and that causes guanine to then base pair with T rather than C. And finally, they can change the structure of DNA, and that's often UV radiation that does that. And when this happens, any thymine that are adjacent to each other base pair up with each other rather than whatever's on the complementary strand, which causes this curvature to occur. So that is mutations. Next week, we will just do chemistry. So let me stop recording.